Hello, everyone, and welcome to today's webinar, Single-Cell Analyses of Checkpoint-Inhibition-Induced Myocarditis, Implications into Mechanisms of Severe Immune-Related Adverse Events. I am Sabrina Lemus of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit www.thermofisher.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credit. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credit. I now present today's speaker, Justin M. Balko, the Associate Professor, Departments of Medicine and Pathology, Microbiology, and Immunology at Vanderbilt University Medical Center. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Balko, welcome. You may now begin your presentation.
Okay, thank you very much for having me. I'm very excited to talk about this subject, which is a research direction that I was not particularly planning on going on in my career, but has been something that we've become very interested in. So my lab in Nashville, Tennessee, at Vanderbilt Ingram Cancer Center is primarily focused on how to make immunotherapies work better in patients through a translational approach. We do this in a couple of different ways. We started off by trying to understand what some of the biomarkers are in patients that might predict response to immunotherapies, because that helps us use these agents more effectively in the clinic. A second thrust of research in our lab is to try to make them work, immunotherapies work better by combining them with other small molecule inhibitors, chemotherapy, or other immunotherapies in order to try to broaden and deepen responses in patients. The last way in which we try to make immunotherapies work better in my laboratory is by trying to understand how to risk mitigate. So just like any therapeutic, immune checkpoint inhibitors or immunotherapies can have significant and severe toxicities, although these differ in their mechanism from many other drugs that we have experience with in treating cancer patients. And so today I'm going to be talking about that last item there, is trying to understand some of the immune-related toxicities that are associated with immunotherapies. So many of you may be familiar, if you're in cancer therapy or simply pay attention to the media, immunotherapies have really transformed cancer therapy over the last 5 to 10 years. So two of these agents, which are approved in a variety of different cancer types, are IgG monoclonal antibodies, which target the PD-1, PD-L1 pathways, as well as the CTLA-4 pathway. So both of these pathways and the understanding that these were checkpoint inhibitors that could alter the outcome of cancer and could be therapeutically employed in cancer patients was really some of the transformative research that won the Nobel Prize just about a year and a half ago. And so both PD-1, PD-L1, and the CTLA-4 pathways are ways to inhibit mechanisms of peripheral tolerance. So both of these molecules in of themselves are ways that our body can inhibit reactivity against cells. So typically CTLA-4 is a ligand which will bind to the B7 family of co-stimulatory receptors on the dendritic cells or professional antigen presenting cells. Although initially this was thought to occur primarily in lymph nodes, we now know that this can also occur in tumor sites or in sites of inflammation. And there's a potential couple other mechanisms in which CTLA-4, inhibition of CTLA-4, might stimulate immune responses. Now, PD-1 and PD-L1, These molecules, the ligand and the receptor being existing on T cells, so the PD-1 molecule shown in the bottom right corner here, at least in the concept or in the context of cancer therapy, can bind either PD-L1 or PD-L2. Although the schematic here shows these molecules present on tumor cells, we now know that they can also be on a variety of other cell types, including other T cells, macrophages, and other stromal cells. And so the interaction between this pathway has the ability to shut off T cells at the site of inflammation. This causes a so-called exhaustion phenotype and limits the functionality of the T cells to do their job, which in this case would be the destruction of tumor cells. Going back to CTLA-4, this interaction in the draining lymph node of the tumor is actually known to, or thought to, at least by one mechanism, limit the co-stimulation and the activation, the initial activation of T cells, which causes them to leave the draining lymph node and seek out the site of inflammation. So both of these mechanisms are presumably purpose in nature for the prevention of autoimmunity and control of acute inflammation. However, we now know that tumors can actually hijack these pathways and use these pathways to avoid anti-tumor immunity. So as I mentioned, while these checkpoint inhibitors, these monoclonal antibodies, can be very efficacious in a variety of tumor types, particularly in combination, with many drugs, including these types of inhibitors, we can have significant toxicities. And what's strange is that, with many small molecule or targeted inhibitors, with many chemotherapies, the toxicities can be very predictable. So we know, for instance, taxanes in cancer therapy have a high degree of peripheral neuropathy, and these have been sort of well characterized. And in many cases, we can control those toxicities, we can pre-medicate, and based on our understanding of how those work. However, toxicity of immune checkpoint inhibitors is widespread and sporadic and can affect a number of different organ systems, shown on the left here. There's also a variety of potential sources of the ways that these inhibitors might affect different organs. Now, the toxicities to immune checkpoint inhibitors are often autoimmune-like, so they induce what are called itises, so inflammation of different organs or organ systems. Our group has had a very strong interest in the mechanisms and the clinical presentation of these different checkpoint inhibitor toxicities. I'll try to consistently refer to them as immune-related adverse events. And you can see in a JAMA Oncology paper that our group published a couple of years ago, where we've looked at across large databases of the World Health Organization's reporting of drug toxicities, and looked at the number of irAEs that were reported versus their fatality rate. What I want to draw your attention to here is, some of the most common ones, colitis, pneumonitis, hepatitis, while they do have a fatality rate, one of the highest fatality rates of any of these, while being very rare, is this third from the bottom one called myocarditis, which essentially means inflammation of the heart. The fatality rate when myocarditis occurs can approach 50% of patients. So this is, while a very rare toxicity occurring in 1% or less of patients treated with the combination of anti-CTLA-4 and anti-PD-1, remains a significant hazard to the deployment of these immunotherapies. So our program at Vanderbilt has started this center called the VPoint Program, which is the Vanderbilt Program for Optimizing Immune Oncology Therapy. This combines a number of different efforts, both from the clinical side, so clinical efforts where we clinically phenotype and organize multi-center projects with collaborating institutions across the country and across the world. We try to institute clinical trials, in which case we either prevent or treat these immune-related adverse events. This also incorporates translational efforts, so systematic collection of samples from patients that are either do have irAEs in response to immune checkpoints or don't, in an overall effort to try to both validate mechanisms correlatively and also identify biomarkers that might predict which patients are most at risk for these immune-related toxicities. We also have basic science efforts, my lab functioning both at the translational and at the basic science side, where we try to explore either distinct mouse models or cell culture models with high dimensional characterization in order to better understand some of these irAEs at the molecular level. So our group, so I kind of got pulled into this way back in 2015, 2016, when a melanoma oncologist is a close collaborator of mine, Douglas Johnson, who runs our melanoma program here at Vanderbilt, and his mentor at the time, Dr. Jess Salzman, who's now at Northwestern in Chicago, as well as Javed Maslay, who are well known for helping to establish the cardio-oncology field, which examines the cardiotoxic effects of many different oncology drugs. And so what happened was Dr. Doug Johnson came into my lab one day and told me that they had two patients in the clinic that were being treated with a combination of anti-PD-1 and anti-CTLA-4 that within 1 week of their first dose and 1 week of one another had both presented into the ER with severe fatigue, very strong cardiac arrhythmias that were not compatible with life. Unfortunately, both of these patients passed away. They were very convinced that this was in response to this combination immune checkpoint blockade, although they had treated a number of patients and had not observed these toxicities. There were some rare case reports from clinical trials and from clinical use across the country that really substantiated that this was likely due to the immune activation secondary to deployment of these checkpoint inhibitors. Our group studied from autopsy samples from each of these patients at a molecular level, both in the heart, in the tumor, as well as the skeletal muscle of the patients. So many of these, or both of these patients also presented with what was called myositis, which was an inflammation of the skeletal muscle, which in some respects can be similar to cardiac muscle and most different from smooth muscle within the body. And so what I want to draw your attention to here was just one figure from that paper in one of the patients where we were looking at T-cell receptors or T-cell clones that we identified by sequencing in that patient's pre-treatment tumor versus the post-treatment tumor, and showing that after treatment, there were a number of shared T-cell receptor clones that were now present both in the tumor as well as in the cardiac muscle at autopsy. What this data suggests, or what these data suggest, is that the treatment with ICI therapy caused an expansion of T cells that were at least found in both the tumor as well as now in the afflicted tissue, which caused the patient's death. Within this paper, we also did some histological analysis just to show the degree of and the consequence and severity of this particular toxicity when it occurs. On the left, you can see a normal heart, so you see these very organized cells and muscle fibers that stretch across. Very few dense purple nuclei. And then on the right, you can see from one of the patients that died of myocarditis, these larger dense nuclei that are lymphocytes. And so these lymphocytes appear to be predominating in areas of the heart where what we have is called myocyte dropout. You can see now this disorganization of the muscle cells within the heart, and that has obviously been some severe consequences and destruction of those cells, which cause death. So immune checkpoint inhibitor-induced myocarditis, what we found here was that it had a high degree of T-cell and macrophage infiltration inside striated muscle. This was associated with electrocardiogram abnormalities and arrhythmias. And overall, through looking at many large databases, we determined that this had about a 1% incidence with combination therapy. The problem is the who and the when. So this is early and unpredictable. And when it occurs, as I mentioned, it can have about a 50% mortality. The combination, there's a combination effect within these patients where often they have what's been described as a triad, a myasthenia gravis-like syndrome, as well as oftentimes concomitant with myositis. This also, although only occurring in about 1% of patients, this presents a larger problem as there are other heart and vessel problems that can be associated with the deployment of these therapeutics. These include other arrhythmias that are not associated with myocarditis, pericarditis, and vasculitis. All of these can have a high mortality rate in patients. So this is some additional immunohistochemistry, just sort of validating this idea that there were a large number of T cells in the heart. Both CD4 and CD8 cells predominate, while plasma cells identified by CD138 are largely negative within the tissue or the affected tissue, as well as just general B cells, so looking at CD20 on the bottom left. Now, there is a large degree of macrophage infiltrate identified here by CD68 immunohistochemistry staining. The importance of this is presently unclear, but something we see very frequently. So one of the first things we wanted to do was get a broader understanding of what was going on within these patient tissues. We initially deployed a strategy called geomics from NanoString. Essentially, this is an in situ technology where tissue sections are labeled with about 40 different protein-based antibodies or protein-targeted antibodies with UV photocleavable barcoded nucleic acid linkers. The tissue is also visualized with several fluorescent antibodies. After staining, one can visualize the tissue architecture with the immunofluorescent antibodies, select different cell populations and or regions of interest on that slide, hit those regions with a UV light which releases the barcodes, and then sip those barcodes off from that region and count them. And so these are just some of the immunofluorescence images. This was from one of the myocarditis specimens. You can see that we have immunostained here with CD3 to mark T cells within the tissue, desmin, which will mark the striated muscle fibers and muscle cells, and then a DAPI stain, which is presented here in false color gray. And so looking across these different 40 markers, without spending too much time on this, one of the things that we did identify was that in these regions with a lot of T cell infiltrate, that we saw a high degree of cytotoxic and memory activated cycling T cell markers. So a high degree of Ki67, a high degree of granzyme B expression within those T cell rich regions, and the expression of the CD45RO marker, which marks memory cells. So we began collaborating with Dr. Jim Allison, Nobel Prize laureate from 2018, his laboratory, who had approached Javed Maslahi, our cardiologist collaborator, where they had these mice in the black 6 background that were heterozygous for CTLA4 and homozygous for loss of PD1, so the gene PDCD1, which encodes PD1. What they noticed was anytime you lost both copies of anti-CTLA4, these mice, regardless of their PD1 status, succumb very early to widespread autoimmunity. In the presence of a single copy or no copies lost of CTLA4, these mice went on to live normal, happy lives. And in the context of heterozygous loss of CTLA4, but complete presence or hemizygous loss of PD1, again, the mice seem to be generally fine. What was interesting was in the context of complete loss of PD1 and heterozygous loss of CTLA4, now looking at the leftmost graph, these mice, the red line here, had a high degree of sudden death syndrome that occurred beginning about 4 to 6 weeks into life. And so in examining pathologically these particular mice and the cause of death, it was determined that these mice were actually dying very specifically of myocarditis, which replicated what we observed clinically in patients who were treated with combinations of therapeutics that target these same molecules. Another interesting finding from the study was that for some reason, the propensity of these mice, which has incomplete penetrance, this phenotype, seemed to be more predominant in female mice, and that's shown at the survival curve on the right, on the left-hand side of the screen. So female mice had a significantly greater death rate. And we actually see this in data from the World Health Organization, where females seem to be slightly more susceptible to ICI-induced myocarditis. These mice also present with a variety of different arrhythmias, which replicate what we observed in human patients. So shown here are some ECGs that have been performed on the mice with loss of PD1 and heterozygous loss of CTLA4, sinus arrest and AV block. You can sort of see normal sinus rhythm in wild-type mice, and in mice with lacking PD1 but having complete presence of the CTLA4 alleles on the left. In the right most plot, we're showing the percent of mice that have conduction disorders. So we do see a small amount of conduction disorders in some of the mice that lack PD-1, but it is nowhere near the amount of those that lack a single copy of CTLA-4 in the context of complete PD-1 loss. There's also this additional bar in this, which I'll get to in a moment. Just remember that treatment with the drug of batacept in that high-risk group of mice seems to ablate these conduction disorders, and we'll get into an explanation of this in a few slides. So getting into some single-cell analysis, we now have this mouse model. It can be very difficult to perform single-cell analysis in patients with myocarditis. The heart tissue is not something that's very easy to sample or to biopsy. And so getting cytotoxicity tissue and being able to do live cell analysis can be an extreme challenge. Occasionally it can be done when a biopsy is taken, but many times these are emergent situations. They're highly dangerous to the patient, and that biopsy tissue is needed for diagnosis of immune checkpoint inhibitor-induced myocarditis. However, we now have this murine model which seems to replicate the clinical phenotype, and so we turn to leveraging this model for single-cell analysis. And so these are some flow cytometry data just showing a very sort of crude analysis here in that the hearts of mice affected, so the PD-1 knockout CTLA-4 heterozygous C57 black 6 mice have a very high degree or enrichment of CD45 positive immune cells within that digested and dissociated heart upon death. So doing a more careful characterization of what these CD45 positive cells are. So looking again, the best comparison here are the red dots versus the blue dots. The gray dots are the wild type mice. You can see that there is, just like what we saw in patients, there is a high degree of heterogeneous infiltration of CD8 and CD4 cells within the context of this particular murine model, as well as CD68. And these were the only markers that we looked at where there was a significant difference between those individual groups. And so it just so happens that these are the three broad phenotypes that we do see, or immunophenotypes that we see in patients that have succumbed to immune checkpoint inhibitor-induced myocarditis. So one of the next things that we wanted to explore was we had this hypothesis that myocarditis in these mice were enriched for oligoclonal T cells. So given that both of these agents act on T cells and are known to induce autoimmunity resulting from autoreactive T cells, we wanted to test the hypothesis that there was an antigen-specific T cell expansion within these mice. So we're able to do this by quantifying TCR recombination and the clonotypes which exist within different environments, most importantly here within the heart of mice that have succumbed to the loss of PD-1 and the heterozygous loss of CTLA-4 resulting in myocarditis. So shown here on the left are the number of hyperexpanded clones, and what I mean by hyperexpanded clones are the T cell receptors in which we, when we sequence, we see those same T cell receptors many, many times in the same tissue. Since presumably one should have a very large and diverse repertoire of T cell receptors existing only a few times within the body, the detection of multiple copies of these same TCRs are generally indicative of the concept of clonal expansion, meaning those T cells have activated in response to an antigen and then have expanded either in lymph nodes or systemically or in the site of toxicity or inflammation. And so what I want you to notice here that in the healthy heart of mice or in wild-type mouse spleens, we see a very high degree of diversity and very low degree of clonality. However, in T cell receptor sequencing from the heart of mice that died of myocarditis within that genetic group that we've been focusing on, you can see that the vast majority of T cell receptors identified there are highly clonal. Now, interestingly, looking across 7 different mice that died of fulminant myocarditis, and also a couple of wild-type C57 black 6 splenic tissue as controls, what you can see is that while there's a very high degree of clonality or T cells that have expanded within those mice, the degree of overlap is extremely low. And in fact, none of those clonal or very oligoclonal T cell receptors identified in one mouse were typically identified in any of the other mice. And this is because this is largely the repertoire within these mice, even though they're genetically matched, is largely stochastic and random. There are some selection processes, but this is also represented on the human level in the knowledge that the T cell repertoire between two genetically identical twins is actually 2% or less of their T-cell receptors. So this actually makes sense that we don't detect the same T-cell receptor. However, this does not refute the hypothesis that a common antigen might be responsible for this phenotype within this model. So looking at this a different way, you can see this is just a Circos plot of the V and J recombination, which is largely dictating the CDR3 or the complementary determining region 3 of the T-cell receptors we identified. On the left is the heart tissue from a fulminant myocarditis mouse, and on the right is the spleen from the same mouse. And so you can see on the right, the spaghetti plot there shows that there's a very, very high degree of diversity. There's very little clonal expansion in the spleen. However, on the left, in the myocarditis, in the heart tissue, you can see that several clones or separate V to J recombination events are highly predominant within that tissue. So now we turn to a alternative single-cell approach, which is single-cell RNA sequencing. So this was a real challenge because you have to have very good viability of your samples to perform single-cell RNA sequencing. In which case it can be very difficult in this model because the mice are fine, they're fine, and then all of a sudden within a 24-hour period, they can get very sick and either die overnight without any indication that they actually have myocarditis. Our lab and Javed's lab have struggled with identifying sort of a biomarker or predictive biomarker of identifying the mice and how to know when they're actually sick, where we'll be able to sacrifice those mice and study the mechanism more closely. However, this was from one mouse that appeared to be very sick, and so we were able to get a live cell dissociation from this mouse. We had to combine 6 different wild-type Black6 mice as comparators in this experiment because of the very low frequency and presence of immune cells within a normal, healthy mouse's heart. And so what I want to draw your attention to on the left is just a UMAP plot where these different islands of cells where the individual cells have been colored, whether they're from the heterozygous CTLA4 PD-1 knockout mouse or a wild-type mouse. You can see that there's general presence of all of these populations in both mice or both groups of mice, remembering again that there were 6 wild-type mice compared to 1 heterozygous mouse. You can see that there's a large number of B cells in the wild-type mouse versus the heterozygous mouse. We believe that that's an artifact of the fact that we combined 6 different hearts in order to do this experiment. We believe this could be just due to inappropriate contamination of just small amounts of peripheral blood due to the processing. What we do see is that there's a very strong sort of phenotypic change in the CD8 compartment within these mice. So that's outlined by the circle here on the left. We also see that there's this sort of new myeloid population, which we've been talking about several times in the presence of that CD68 population that we observe in the hearts of both human and mice suffering from myocarditis. And so what was interesting is we also had single-cell level T-cell receptor sequencing data on these same single cells. So we're now able to look at those clonal TCRs. We're able to match alpha and beta chains of the T-cell receptors. And we're able to separate in that heterozygous myocarditis mouse's heart the T-cell receptors based on whether they were clonal, meaning clonally expanded, or whether they existed only a single time within our single-cell RNA-seq data. And what I want you to appreciate here in the middle plot is that the vast majority of these clonal T cells express CD8, and they do not express CD4. So this experiment prompted us to look a little bit deeper and to say, okay, so is the myocarditis that we're observing in these mice, is this a CD8 or a CD4 mediated effect? So you can see in the survival curve that with no treatment, about 50% of these mice succumb to myocarditis within about 2 to 3 months after weaning. With the administration of depleting antibodies to CD4, we see no change in that survival. However, when we deplete CD8 cells, we see a near abrogation of mortality due to myocarditis. So, looking at the predominant T-cell receptor alpha and beta chain within our single-cell sequencing data, we're able to combine those, that predominant clone, which was highly expressed within the heart tissue of that one mouse that we did single-cell analysis on. And what we were able to do is do a computational analysis across what's known about the B allele for MHC class 1, now knowing that this is a class 1 restricted antigen, and looking at things that are expressed specifically in heart tissue versus other organs. And so doing a computational approach and then recombining both the alpha and the beta alleles in a recombinant TCR and then transducing a murine CD8 T cell line called the 5kC T cell line, we're able to reconstitute that T cell receptor. And we now have a model to study antigen specificity of that T cell receptor. As a control, we reconstituted the OT1 T cell receptor, which recognizes the synfecal antigen. So these 5kC cells will express interleukin-2 in response to the appropriate antigenic stimulation. We're using bone marrow-derived dendritic cells from a C57 black 6 mouse in order to perform this experiment. And when co-cultured with an OT1 T cell receptor expressing T cells, those bone marrow-derived dendritic cells respond only by IL-2 secretion only to that ovalbumin peptide synfecal, and that's shown in the upper right here. Now, in that C1 clone, which is that very predominant T cell receptor, which we reconstituted in similar cells, we see that the predicted antigen from a cardiac-specific transcript is now inducing the expression of interleukin-2, specifically within the T cells that express that clone. So we now think we know at least some potential idea of what the cardiac antigen is, in which case loss of PD-1 and CTLA-4 is now releasing peripheral tolerance against. So given that these findings appear to be at least partially dependent on haploinsufficiency of anti-CTLA-4, collaborators of ours in France, in which case that had a patient with myocarditis last year, published this letter in New England Journal of Medicine, where they hypothesized that given no other options in this particular patient who was very clearly going to die from immune checkpoint inhibitor-induced myocarditis, that the administration of a drug called the batacept, which is the CTLA-4 molecule fused to the Fc region of IgG1 for therapeutic administration, often used in transplant rejection, might reverse this phenotype or this toxicity. And so I encourage you to go out and read this letter. It's a very short read, very interesting if you're clinically sort of oriented. This patient had a very rapid recovery with administration of a batacept, and after about a week was able to leave the hospital and continue on in their life. And so we hypothesized that perhaps if representative of this clinical scenario, that mice treated with a batacept, given the fact that there is this propensity of these mice to develop checkpoint inhibitor-induced myocarditis, just like combination treated patients, that a batacept might reverse this phenotype. So just like we saw in our CD8 depletion, the administration of a batacept to this group of mice that lacked PD-1 and were heterozygous for anti-CTLA-4, specifically in the females that were most susceptible to myocarditis-induced death. You can see that this massively reversed this phenotype, and these mice were now able to survive. So in looking at the CD45 positive infiltration within the cardiac tissue, again, we can now see that there's, in the vehicle-treated mice, there's this enrichment of CD45 positive infiltrating cells within the heart tissue. This takes a little while to go down. So in about 2 weeks, we don't really see any difference in the absolute number of CD45 positive cells within that tissue. However, by 14 weeks out, we're able to see that these mice now look like wild-type C57 black 6 mice. So this replicates at least this one case study that was published, that a batacept might be a suitable treatment for this myocarditis when it occurs. So we wanted to study this on a little bit more mechanistic basis. We are certainly still looking at how common this myocarditis-inducing antigen is in the cardiac tissue in the mice. How common is it among different mice? Is it able to, is immunization with that peptide capable of causing myocarditis in the black 6 model? And what are the possibilities that that antigen is similar or the same, at least in its protein source, in human patients? However, we wanted to understand a little bit more about these T cells on an immune phenotyping backbone or background. or context. And so we employed cytoff or mass cytometry on the cardiac infiltrating cells. So this is just another example of single cell analysis that we've performed in order to answer our questions. This is a Tisney plot. You can see where we're able to identify all the different predominant immune populations. Again, the most predominant one being CD8 positive T cells, showing the upper right of the 6-panel quadrant in the bottom left corner of the screen, and then also high constituents of CD11B positive cells, our myeloid cells, and our CD4 positive cells. So this cytoff data replicates what we saw by single cell sequencing, what we saw by standard flow cytometry. We also had this one really interesting finding within these data. So looking at CD86, which, if you remember right, is one of the two co-stimulatory ligands for CD28 on T cells, but also that is blocked by CTLA4. And so presumably a drug like a batacept would directly bind to CD86 as well as CD80. Those can occur on professional antigen-presenting cells. We're actually quite interested to see that these CD8 cells that we see within the myocardium, or infiltrating cells within the myocardium, was highly present. CD86 was highly present on those CD8 cells. So it's not really well understood what a co-stimulatory molecule might be doing on T cells within an inflammatory microenvironment, although it is well known that interleukin-2 can induce in sites of inflammation the expression of CD86, but not its partner CD80 on T cells and other cells within an immune microenvironment. And so we've actually started to explore this in the lab as a possible alternative mechanism for the action of a batacept. So is it possible that CD86 on those T cells might be a direct target of a batacept? And so we're now trying to understand whether CD86 expression on T cells can either stimulate in cis or in trans other T cells, and whether a batacept's functionality within at least this model and or human patients might be associated with its ability to bind CD86, shown here on an APC, but now we know also expressed on T cells within this microenvironment. And this, we're hypothesizing, could be a potential mechanism because of this rapid recovery that we see whenever we treat either mice or in some cases humans with a batacept, which isn't consistent with an already primed T cell response secondary to the administration of anti-CTLA4. So in conclusions, we know that myocarditis can be caused by immune checkpoint inhibition. This can be associated with T cell expansion and a phenotype of memory activated CD8 positive T cells, at least within this model that we're looking at here, as well as in some human patients. In mice, genetic loss of PD1 requires heterozygous loss of anti-CTLA4 in order to induce myocarditis that seems to approximate that which we observe in patients. Myocarditis in mice seems to be associated with oligoclonal expansion of private TCRs in each individual mouse, and in humans. We have some data there, which we haven't presented here today. But in general, at least in mice, this seems to be predominantly CD8. We're now looking at trying to explore how likely it is that it's either CD8 or CD4 across human patients as well, in order to see how well we think we're actually recapitulating the etiology and pathogenesis in human patients. In both mice and humans, the delivery of a batacept appears to reverse or protect against myocarditis and cardiac inflammation due to loss or inhibition of these immune checkpoints. And we're now exploring CD86 possible T cells as a possible mechanism of action of a batacept. Finally, I showed you sort of some provocative but early data that we might have identified the target antigen in mice and humans, but are really holding off on reporting on this until we're convinced that this wasn't just a single mouse, that it's consistent among mice, and whether this is translatable to the target antigens and the parallels there in humans. And so with that, I want to acknowledge, many people. This isn't all our laboratory's work. In fact, we're just really performing a small but integral part of this. Our funding is by a variety of sources, but specifically for this project, two main sources: the National Institutes of Health, both Dr. Moslehi, Javed Moslehi, and myself and Doug Johnson, have several grants within this area, either specifically studying myocarditis. Doug Johnson and I have an NIH provocative questions grant that is focused on identifying the mechanisms and biomarkers of immune-related adverse events in general, secondary to checkpoint inhibitor therapies. The number of people in my lab who we like to call the cardiac kids, as well as in Javed's lab, who would stay up late and be processing these samples either from clinical patients or from mice whenever the unexpected findings would occur, that we would need to suddenly have a big mouse takedown day or have new clinical samples that needed to be processed immediately. And so obviously we couldn't do that without their expertise and excitement and enthusiasm. A number of different clinical pathologists we work with, as well as Tcell biologists. And then finally, the model, which has provided a lot of the insight into this, provided by James Allison's lab at MD Anderson and his postdoc, Spencer Way, who initially described this finding. And so our lab is working closely with theirs to try to translate some of these findings to helping patients that are being treated with these immune therapies and to better understand these immune toxicities when they occur. We also had some support for NanoString in order to deploy some of their spatial profiling techniques, which I think was very helpful in us sort of driving our more specific single cell analysis techniques downstream. So with that, I'd be happy to open it up to any questions the audience might have.
Thank you, Dr. Balko, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. Let's get started. Our first question, Dr. Balko, is what are some ways that single-cell analyses have really provided a unique opportunity to answer your research questions in this project?
Sure. So that's a great question. And as you know, the immune system can be highly complex. And as soon as you have an initial inflammation or insult, oftentimes what we observe is sort of this mass flooding or sequential flooding of that tissue with all different components of the innate and adaptive immune system. And so sorting out what's really going on there is a very complex technique. Many times, the pathogenesis of immune-related toxicities, of autoimmunity, or even an anti-tumor immunity can be dependent on very small populations of cells that you might not be looking for. You might need a large number, of different markers in order to really identify those cells and to get a better understanding on what's going on, either transcriptionally or phenotyping within that population of cells. I don't think that we could do this type of analysis or really answer this research project or question without being able to employ those technologies that allow us to very quickly and in a facile fashion figure out what's different, what's going on, and how are those cells changing, how are they adapting to that environment, how can we answer that question without looking at a single cell level. And the last sort of answer to that would be the T-cell receptor sequencing and our single cell RNA sequencing. In order to understand what the T-cell receptors are and what they might be recognizing, one has to be able to link two different genes, the alpha and the beta receptors, or the alpha and beta chains for the T-cell receptors, together in order to know what that pairing is. And in a mishmash of RNA sequencing data or even T-cell receptor sequencing data, without looking at the single cell level, there's simply no real confident way in order to pair those alpha and beta chains, localize them to the same cell, because you require both of those chains to be sequenced in the same cell in order to dictate what its CDR3 region actually is.
Wonderful. Thank you, Dr. Balko. Our next question is, what is the major impact of this work? For example, how can knowing the mechanism of ICI-induced myocarditis lead to changes in the way we treat patients?
Yeah, I think that the major impact is pretty broad. I hope that we're able to completely figure out this mechanism. I'm confident that we will. I think it's easiest to answer that in the context of examples. So if we have a common antigen, for instance, that's present in these patients, oftentimes what you see are B cell antibodies that are produced in response to inflammation in a certain tissue, and many times those B cell antibodies in the context of autoimmunity can be the same or similar as what the T cell antigen is. And so, just as one example, is it possible that some of these patients already have some degree of autoimmunity against the heart? And if we're able to figure out what that antigen is, can we identify, for instance, the presence of autoantibodies in those patients? If we're able to fully characterize the T cell receptor repertoire, can we identify patients that might be most at risk because they have either the right HLA types or the right T cell receptor repertoires already existing prior to being treated with immune checkpoint inhibitor therapy that put them at risk? So can we risk mitigate? Can we identify those patients up front? Can we identify them early? And then sort of a final question, or a final possibility along those lines are, could we, if we know the antigen, could we generate, for instance, tolerogenic vaccines? So vaccines that help to generate regulatory T cells or other immunosuppressive arms of the immune system against that particular antigen in order to protect that patient while still allowing them to be administered this highly active combination of immunotherapies.
Thank you again, Dr. Balko, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, stay healthy. Goodbye.